Okay, so welcome everyone. We are happy to meet again for another great talk of our CASP RNA special interest group, once again about application of AI, but not only. So I will introduce our today's speaker. So it's Jianyi Yang, and is a distinguished professor in Shandong University of China. His research interests include protein RNA structure prediction. He's made significant contributions to the development of a few widely used tools, including trRosetta and iTaser. And his group won the tertiary structure prediction of protein in CASP 15. And he has published over 70 papers that are cited over 15,000 times. And after his talk, his PhD student, Wenkai Wang, will present the results about CASP 15 for RNA. So yeah, with that, the screen is yours.
Okay.
Yes. Yeah.
Okay. Thank you, Martin, for the nice introduction and the invitation to this CASP special interest group. So I'm honored to be able to share our work in this meeting. So as Martin said, after my presentation, my student Wenkai will introduce some detailed results from the CASP 15 modeling. So my talk is for some overall view of our method. So I'm going to start. So basically, my talk has three parts. The first one is a brief introduction about why we developed the trRosettaRNA. So because previously we are mainly focused on the protein structure prediction, so I want to briefly talk about this. So for this popular problem of the protein structure prediction, the task is to find a function y equals to fx to map this sequence x into this 3D structure y. This, well, everybody knows this is a very difficult problem. It's a grand challenge. But recently, this was almost solved, especially for this single domain protein structure by AlphaFold 2. Of course, before AlphaFold 2, there are many other works, including our own trRosetta in collaboration with David Baker and also others. So this AI-based method is to minimize the loss of the predicted label fx and the true label y0. So to make to do this minimization, we need data to train the network F. So this is the big data for protein structure prediction. First, as we said, the mathematical model is y equals to fx, and this first variable, the x, is the protein sequence. By the DNA sequencing, we have more than 200 million sequences uniportal databases. So we can use this data to find the multiple sequence alignments to encode the co-evolutionary information in the protein sequence. And for this Y, we have this protein data bank, PDB. So this data bank has more than 0.2 million structures. So this data can be used as a big data for training this network F. Then how exactly we use this network to solve this problem, the AI to solve this problem. So from the input protein sequence, we are using some sequence alignment method called attributes or M62 to find a homologous sequence to build this multiple sequence alignment, in brief, MSA. And this MSA was input to the network, shown here. And so this network is just illustration. You can use any kind of network, such as ResNet and also the transformer used in AlphaFold 2. And we know this from this network, our objective is our goal is to predict the structure directly. Unfortunately, in 45 years ago, this seems to be difficult. So people are trying to break this problem down, make it easier. So you can see here, I call this a two-step approach. So this network is trying to not predict the 3D structure, but just to predict a 2D image. So this is a simple representation of the 3D structure. Then the second step is to recover from this 2D image to the 3D structure. So this kind of approach, there are two representative, for the most representative ones. Probably the first one is AlphaFold 1, and inspired by the AlphaFold 1, we developed the trRosetta. But probably the problem with this kind of two-step approach is probably the accuracy is limited. So to improve this two-step approach, then ideally we can predict the structure directly from the 3D structure directly from the neural network. So that's called the end-to-end approach. And the representative method for this end-to-end approach is AlphaFold 2 and also RosettaFold. So our group we developed trRosetta. Then we want to continue developing this kind of method. You can see this is the trRosetta. The idea is to here is to improve the label Y. As I said, the network is at the beginning it's difficult to make the network predict 3D structure. So we break it into this image. So in this work, so by collaborating with David Baker, we find some besides this interresidual distance, this first image shown here is the interresidual distance. We use other orientations to approximate this 2D image, not just one. We have, in fact, here we have six different 2D representations. In fact, because two of them are symmetrical, another two are not symmetrical, so we got in fact 6 different variables for this Y. So this is probably the story about the protein structure predicting by our group using this trRosetta. So after this work is done, so we want to move to the RNA structure prediction. Probably most because the AlphaFold 2 already make the protein structure prediction very precise. Then how about the RNA structure prediction? So based on our previous work, we are trying to extend it to this RNA structure prediction. So this is the second part of my talk, trRosettaRNA. So as I said, this trRosettaRNA is an extension of trRosetta. So it's a two-step approach. So before that one, I want to talk about the data. So as I said, the network need much data to train. Then how about the RNA? For all of you know that the PDB data bank, they have this, the gray part is the protein structure. It's more than 19%. However, for this nucleic acids, the data is limited. So for nucleic acids, we have DNA and RNA. Also, most of them are complex with protein. So by taking these two parts together, we can get about 80% for this nucleic acid structure. But for RNA, so it's much more limited. It's about in total now it's about 7,000. So it's very limited. And the problem is these data are very redundant. After you remove the redundancy, you get only a few hundreds non-redundant version. So it's very limited RNA structure data. But we can also try to see whether deep learning can be used a lot for this RNA structure prediction, mostly inspired by the protein structure prediction problem. So in fact, our group tried to studied this problem a few years ago when we are studying the protein inter-residue contact prediction. So after that, we just try to see whether this deep learning can be used or not. So this is, I think this is the first deep learning based approach to this RNA internucleotide contact prediction. So this work was published only a few years ago. You can see this network is very simple. It's just a very shallow resonant block to predict this image. This is a 2D inter-residue internucleotide contact. So in our test, we have two different data sets compared with this work published in Cell. You can see this is the precision of a predicted contact in comparison with this PLMC. This is the direct coupling based method. You can see the predicted contact, the precision of the predicted contact by our method, we call it RNA contact, is all better than this direct coupling based method. So on average, it's about 2 times higher than this DCA based method. Unfortunately, when we test whether this kind of predicted contact can be used to improve the 3D structure prediction or not, so we are disappointed to see that the improvement is not very significant. So we just try to, okay, let's just go to the 3D structure prediction directly. So that's the method we call trRosettaRNA. So this is after the development of trRosetta. You can see here the pipeline is similar to trRosetta in the methodology. So basically, it's a two-step approach from this input RNA sequence. So we want to predict this 3D structure, but it's okay. We just we don't have enough computer hardware to train an end-to-end approach. So here we just use this two-step approach. From this sequence, we are predicting this 1D and 2D geometries, and the second step is just to recover the 3D structure from this 2D images. So this is the overall idea of the trRNA, and in detail, you can see here the first step is still similar to the protein structure prediction. From the sequence, we are trying to find some homologous sequence to build this MSA. Only one slight difference compared with the protein structure prediction with this input, the secondary structure. In fact, in the RNA structure prediction community, the RNA structure, secondary structure is very important. So this is very different from the protein secondary structure because this is a two-dimensional relations, but for protein it's just a one-dimensional data. So in our method, we are using the sport deep learning based segment structure prediction. It's just sport RNA to predict the segment structure. And here the network is just borrowed from the AlphaFold 2, the A-formal network. This is a transformer-based neural network to predict these 2D image and 1D images. So this is the pipeline of our method. This figure B is just this network, the one block of this network. So I'll just make this brief introduction about our method. And also we developed this web server in our lab website. So you can download everything here, the source code and the training and the test data, everything there. And also you can submit the RNA sequence to this server and you will get the results shortly. So this is the method of trRosettaRNA. So after this development, we need to make a rigorous validation or test of our method. Basically, we tested our method with 4 different data sets. The first two is independent test. The last two is blind test. So I will talk about the overall results of this test in my next slide. First is an independent test on 30 long redundant RNAs. So compared with automated prediction by conventional methods, here we are using RNA and uncomposer and control. You can see by this automated prediction, this figure shows that our method can predict for most targets with lower RMSD than these two methods. So on average, it's about improved by about 2 times compared with the automated method. So this is the first test. Another independent test is based on the RNA puzzle targets. So here we are collected 20 targets from RNA puzzles. In this experiment, we show so the radio test group is one of the top groups in this RNA puzzle experiments. So we compared with this group. You can see that here our method make the automated prediction. So it's about putting slightly worse than this test group. We want to claim that this is based on that's fully automated prediction. But this is just the intent test. Probably a more rigorous one is based on the blind test. So that's the next two tests I want to show. So the first is based on this one, probably everybody already know. So in this CASP 15, there are 12 RNAs. You can see this left hand side, we have 8 natural RNA. On the right hand side, there are 4 synthetic RNA, recombinant RNA. You can see there are some so here the gray structure is native one, and the red cartoon is predicted models by our method. Here, this figure, we are taking the best submitted model from the 5 submitted ones. So you can see here the designed targets. So you can see these RNB, the designed targets has probably higher RMSD, means we predict this designed RNAs badly than these natural RNAs. So I want to show you the ranking for these two different type of targets. So for this ranking just based on the RMSD of the Z-score of the RMSD. So you can see for this natural RNA, our method is ranking at the second by this RMSD-based ranking. So the top one here is actually another method based on the conventional method. However, for this synthetic RNA, the ranking so it's much lower compared with the natural RNA. You can see it here. It's this one. So it's very bad for this synthetic RNA. So here I want to make something. I make a comment. For this synthetic RNA, so you can see here I have 2 different colors, red and blue for both figures. So the red bar are from the automatic prediction, and for the blue bar they are the human group. So here, the top probably overall also the top 4 are based on conventional physical-based method. Other pretty deep learning based, not just our group. There are few other groups in the world with deep learning methods. So you can see here the top groups are not using deep learning, but I don't think this is a reason. This can explain the deep learning is worse than the conventional methods, and they already show in the first 2 independent tests, especially the first one. The deep learning-based method can improve over the conventional method on the RNA test. But here you can have different signal. But in my opinion, I think this is because the top 4 groups or the top groups for this synthetic RNA manually are human groups. They are using human beings to improve the prediction manually. So that's I think that's the real difference between these top groups and other, I mean the blue groups and the red groups. So this is the first test, the blind test in CASP 15. So in fact, at the end, I will show you some more evidence about my conclusion. So another blind test is in RNA puzzles. In RNA puzzle, this is an ongoing experiment. So we also participated this experiment after we developed the trRNA. So now there are 3 targets with results summarized on the web page. So you can see for these first 2, we have predicted the structure reasonably with this RNA about 10 angstrom. And also the ranking of our prediction is also acceptable. You can see it's just after 2 human-based groups. And for this one, we didn't do very well, and I try to find a reason for this. This is because there are no similar structural sequence to our training data set. So this is another blind test results. So I want to see that this since the automated prediction is promising, no, it's not very, not yet very accurate. So I want to see, conclude why what can make a prediction, automated prediction difficult for RNA structure prediction. So here is the analysis about the previous 4 different tests. So you can see here, we're using this metric to measure the difficulty. So this is the TM score of the tested RNA against the existing data in the protein data, existing RNA structure data in PDB. You can see here this score for these 4 different tests. So for the 30 RNAs, independent RNAs, you can see this TM score to the previous RNA is very high compared to other 3 data sets. And also, if you remember, for this data set, our honesty for this data set is about I don't remember. You can see this one. So it's our 8.5 angstrom. But for this, you can see for this partial data set, it's about 10 angstrom. Means it becomes harder. And this this can be explained by this increased this difficulty from the blue increased to decreased to this. I mean, the TM score decrease means the difficulty increase. So from this blue to the green, you get the targets becomes harder. And also for this, you can see the honesty of our prediction gets higher. So it means this match explains a lot, in my opinion. Also, similarly for RNA, for this CASP, and for these 2 blind test targets. So this is the reason why what can make the automated prediction difficult. So because this is based on the deep learning methods, they are based on the training. Training means you need to have some similarity between your training set and your test set. So if the similarity becomes lower, then you can anticipate the performance will decrease. I think that's probably that's the reason. And especially for this CASP15, these synthetic RNAs, so why they are so difficult to be predicted for this AI-based methods. So we argue that we need a human intervention, human assistance to improve the prediction. So I have some evidence here because the top 3 groups in CASP15 predictions, CASP15 experiments already have their paper published online. So I read the paper clearly and I find some clue. You can see here. This is the first group in the CASP15 experiment. You can see in the abstract, they said this sentence. They say they use the manual verified resource for predicted signal structure. Also, in the conclusion, they say the manual intervention is necessary for most cases. So this is the first group in the CASP15. And another group is the Chen. So this is the address of the paper. You can read it online. So in their paper, they also said they need a manual intervention to predict the long-range contacts for this, especially for this human-designed targets. Also, the third group, this RNA policy. So they also write, we need manually modified to ensure repeated strand motif, etc. So that's, I think that support my previous conclusion. So for this, for very hard RNA targets, so it's very difficult to make an accurate prediction for automatically. By I think by any method, not just for AI approach, for conventional method, this is also very difficult. So I think this okay, it's totally okay. I'll just stop here for this slide. So I think that for my last slides, I will talk about the future direction for this RNA structure prediction by deep learning based methods. So as I said, the top ranking groups in CASP 15, they are better than AI-based method. But I think the reason is because they are using human intervention, not just because AI is not good. Then how to improve AI in future? It's also based on this brief function y equals to fx. So in this function, we have three parts, y, f, and x. Then let's see how about these three parts in future. First, for the input part of the x, the sequence. First, for the RNA is very difficult to make accurate MSA. Sometimes you cannot find the homologous sequence, so it's hard to build MSA. And even you find homologous sequences, it's so hard to build accurate MSA. So it's different from protein. Then how can we remove this part? We can just build a pre-trained RNA language model. So we already see that there are some models published online, or some do build models, but in fact I will try some, but do not work very well at this stage. But we hope that in future we can make it better pre-trained RNA language model to solve this first problem. Another one is secondary structure. As we said, our method rely on secondary structure prediction by stored RNA, but sometimes this different based method can also make a completely wrong prediction. So it means if we can have better secondary structure prediction, we can also maybe improve the 3D structure prediction. So the last one is to have some experiment data to assist the modeling. So then another variable is y. So this one, so we have the problem is, but this is the most severe problem. We don't have enough training data for this deep learning based method, especially this label. So what can we do with the current existing data? Well, we can try to say break the structure into fragments and reuse the samples, especially for some very big RNAs with more than 1,000 residues. Only this can be helped to some extent. Another one is use the protein data. We can try to predict the protein RNA complex. Then also some RNA cannot form stable structure. They need to bind the protein. So this is also one way to improve. So the next one is function app. So we can use AI and create more powerful AI, more powerful network, plus this physics-based predictions. So as I said, so the CASP 15, the top groups are using physics-based. So it means physics-based modeling can be also useful for to be included in AI-based predictions. So because we anticipated our method to be automated as automated as more automated is welcome. So many people, more people can use the method. Okay, I think I want to stop here. I want to thank my David Baker during the development of trRosettaRNA in his lab, and also my lab member. So I've got here my student Wenkai will talk about some results about the RNA structure modeling. Okay, I will stop here my screen.
Okay, but maybe we have some questions before we go to the second part.
Okay.
I don't know if we have any questions, but yeah, now because I have one. I think what you mentioned that for now your method is not end-to-end. I think what you said is like too expensive to build end-to-end method for your method. So now it works more like AlphaFold. So it predicts orientation maps or distance maps. I don't remember exactly. But based on that, you do modeling. You think the end-to-end model or the end-to-end architecture would be also helpful in this case, and how expensive it is?
Okay, thank you for the question, Martin. So currently we are not end-to-end, we're just a two-step approach, just very similar to trRosetta. So the reason is probably the major first probably the reason before is based on this hardware. So we don't have enough resource to train an end-to-end approach. So because the 2D image is much easier to train, so this is the major reason. And with this pipeline, so it's very easy to train. We just use one, I think A100 card is enough to train a model in less than a week. So it's very cheap. But for end-to-end, because we get a better hardware recently, so we try to use the end-to-end approach to improve. But unfortunately, we didn't find any significant improvement. So then we guess the reason is still the same because the data is too limited. So the end-to-end approach, they need even more data to train. So it does not help enough. Okay, that's my answer.
Yeah. So it's interesting because we need more data to train end-to-end, and at the same time, end-to-end is very expensive. So then we need even more resources. But yeah, I see your point. I don't know if we have more questions. I don't see it. So maybe we'll have the session for questions also at the end. So yeah, maybe we can now go to the second part. So now a PhD student, Wenkai Wang, will talk about the results for RNA CASP.
Okay.
The screen is yours. Yeah.
Can you hear me?
Yes, we hear you well. Yeah.
Okay. My name is Wenkai Wang. I'm a PhD student at Nankai University and supervised by Professor Jianyi. I will share some details on our trRosettaRNA method in the CASP 15 computation. Here is the overview of trRosettaRNA pipeline. It has been introduced by my supervisor. So I will only give a short description of the RNA former pair. This RNA former is largely borrowed from the RNA former in AlphaFold 2. That is to interactively update the MSA representation and the pair representation with the attention modules. The main modification here is that we use a multi-scale convolutional module named REST2net to assist the channel update. This can enhance the ability of the network to model the local details. Here is the definition of geometries we want to predict, including the 2D distances, 2D orientations, and 1D orientations. Here, the 2D means the relationship between any pair of nucleotides, for example, between the I and J. And 1D means the relationship between neighboring residues, for example, between I minus 1I and I plus 1. Similar to trRosetta, we divide the value range into tens of bins, and we predict the probability of the value falling into each bin rather than to predict the real value. So this is a multi-class classification test, and the loss function is cross entropy. After the neural network, we transform the predicted distribution of geometries into energy potentials. These potentials exhibit a negative correlation with the probability values. And starting from a randomly initial structure, we employ the LBFGS algorithm to refine these structures by minimizing the total energy. This results in a 4-atom decoy. And to improve the stability, we generate 20 initial structures for each target, and yielding 20 decoys. The decoys with the lowest energy is chosen as the final prediction. We used two training sets to train the RNA former network. The first consists of over 3,000 RNAs from the PDB database and were released before 2022. These RNAs both have at least one homologous sequence so that the MSC can be used. The second is also borrowed from R42, is that we established a self-distillation dataset from the BPRNA. And these RNAs have annotated secondary structures, but their 3D structures were unknown. During training, we firstly train a network on the PDB dataset, and then we generate self-distillation labels, meaning we predict geometries using the pre-trained model. Finally, we further trained the model on both two training sets with a 1:3 proportion between the PDB dataset and the self-distillation dataset. For each target, we also provide an estimation of model accuracy, namely ERMSD, meaning the estimated RMSD. This estimation mainly considers the convergence of the top decoys and the confidence of the predicted distance map. Here we can see that this estimation correlates well with the real RMSD value with a PCC of over 0.5. Based on trRosettaRNA, we participated in the CASP 15 competition as a server group named the Young Server. For its target, we generate two MSCs. One is using the RMSC tool, and the another using the Inferno tool. And we selected the MSC with the lower EMSD to fit into our network. The secondary structures were both predicted by spot RNA. We did not investigate other secondary structure during the competition. However, I will later discuss some cases where this point can be improved by exploring other secondary structures. For each target, we generate 20 decoys and we submit the 5 decoys with the lowest energy. As mentioned by Professor Yang, our groups ranked at the 9th position in the official ranking and the 5th position in terms of the RMSD. For the 8 nature RNAs, the RMSD of our group was overly surpassed by the leading human group AHMRN2. And we can see that there were only 3 targets where our group performed worse than the top 2 human groups, AHMRN2 and the Chen group. This reflects that for these nature RNAs, our fully automated method can be competitive with the leading human groups without any human intervention. Here is a case where our prediction is more accurate than the AHMRN2 model. That is the R1116. This figure shows the DCA matrix extracted from the MSA. We can see that it exhibits a similar pattern to the real contact map, which means that this MSA contains most of the correct interaction information in the 3D structures. With the help of this MSA, our prediction is more accurate than the predictions made without MSA. There are 4 natural RNAs for which young server modules are worse than the ARSAMI RNA2 modules. We found that for 3 out of these 4 targets, the performance gap is mainly caused by the input secondary structure. As mentioned before, all the young server modules were generated using the secondary structure predicted by sport RNA. However, after the computation, we found that for these 3 targets, the template secondary structure can be easily detected from RFARM database. For example, the sequence of R1107 belongs to the RFARM family 622. We can easily obtain a template secondary structure from this family using an automated protocol named R2DT. Note that the RFARM database used here was released in 2021, so there is no risk of data leaking. And this secondary structure is more accurate than that predicted by sport RNA. The corresponding 3D structures is also much more accurate than the submitted young server modules, with the RMSD drops from 17 angstroms to 4 angstroms and is competitive with the model from the top human group RNA2. We also noticed that the EMSD can capture this improvement caused by the secondary structure. This reflects the potential for further enhancement of our method to integrate more secondary structures if possible. A similar trend can also be observed from another target R1108, which is also from RFARM 622. This helps drop the RMSD from 9 angstroms to 4.8 angstroms, and the EMSD also shows the same trend. For the R1149 from RFARM 3120, we can also observe a similar comparison. R1156 is the overlay case of natural RNAs where our method cannot produce competitive results, even with more accurate secondary structure as input. This RNA lacks any homologous sequence of structures in the current database. We can see that modeling such RNAs is still challenging for all deep learning-based methods. The best model from the deep learning methods has an RMSD value of over 12 angstroms compared to 5 angstroms from the human group. So in summary, for these natural RNAs, our automated method is competitive with the top human groups, even without any human interventions. The usage of MMC helps improve the performance, and further enhancement can be achieved with better secondary structures. However, we admit that the accuracy is still far from satisfactory with the average MC of over 10 angstroms, and especially for the no RNAs, which has no similar sequences of structures. But we believe that this issue can be addressed with the increase of RNA data in the PDB database. For the remaining 4 synthetic RNAs, both our ranking and the MSD values were much worse than the leading human groups. That's like Professor Yang introduced. We think that this bad performance can be attributed to 2 reasons. Firstly, similar to the natural targets R1156, these synthetic RNAs lack homologous sequences and or structures in the database. The left figure shows the relationship between the RMSD of our predictions and the structure similarity to the known RNAs in the PDB database, we can see an obvious negative correlation here. This means that our method may perform better on those RNAs that have similar structures in the PDB database. However, the four synthetic RNAs exhibit lowest level of structural similarity to non-RNAs. This partly explains the poor performance of our method on these special targets. Secondly, we found that it is challenging to model these RNAs in a fully automated manner, regardless of whether using traditional methods like CMRA and RNA transposer, or using deep learning-based methods like RuFold and our trRosettaRNA method. Just like Professor Yang said, as mentioned in the paper by the top human groups, the accurate modeling of these RNAs needs human interventions, such as manually identifying long-range interactions and assigning some certain structural motifs. Therefore, we conclude that the performance gap mainly comes from the difference between automated and human intervened modeling, rather than from the difference between the deep learning-based and the traditional methods. In fact, both methods perform closely in terms of the automated modeling. Here we use target R1138 as an example to illustrate the challenges of automated modeling of these synthetic RNAs. We can see this circled tracing loop in the native structure was missed by all automated methods in the predicted 3D structures. Actually, if we look at the distance map predicted by trRosettaRNA, which is displayed here, we can see that our method successfully captured these long-range interactions in the predicted distance map. And it is easy to obtain an accurate tracing loop motif if we only use this part as the reference. However, this tracing loop was missed when modeling the global structure. We think this is probably due to the completed interactions from the other part of this structure, which makes it difficult to accommodate this tracing loop if there were no human intervention. So we think it may be promising to combine deep learning with the traditional fragment assembly methods when modeling such complicated synthetic RNAs. This is one of the directions we plan to explore in the future. So in summary, for these synthetic RNAs, they have no homologous sequences or structures and were difficult to automate modeling. However, our approach maintains competitive performance with the traditional methods under the constraint of full automation. And we can correctly predict some motifs in the distance maps. So these motifs were not reflected in the tertiary structure. So as my supervisor, Professor Yang, just mentioned, inspired by these findings, we are working on further improving our method in various aspects. We hope that more advanced deep learning methods can be developed in the future. Finally, I would like to thank the help from my collaborator Chen Jie Feng and my supervisor Professor Yang. And I'd also like to extend my thanks to the CASP SIZ group for giving me the opportunity to talk here. So thanks.
Thank you. So maybe I will start. There is a question, but I will start maybe because we just mentioned, but I will have a question for both of you. So what are your thoughts? What would be the best way to tackle the problem with synthetic RNAs? Like, do you have any ideas how to actually model synthetic RNAs? Because they will probably come in new CASP.
Okay, so thanks for question. So for this synthetic RNA, so it's really a challenge for AI-based methods, also for other methods. So for this probably I think I don't know if the human intervention is necessary or not. At least in our example, in this CASP example, it's human intervention is needed. So especially one can already show a few examples. If you have a better predicted secondary structure, then you can definitely improve the AI-based predictions. So also for this, yes, these examples. So also I read other probably top groups in CASP 15. So they are also trying to improve the secondary structure prediction by some are reading literature to collect information or just correcting the long range contact by experience. So I don't know for this automated prediction for synthetic RNA. I think it's hard to solve the problem unless we have much more data, training data. But I don't know if by myself, I don't know it's very different from the protein design. So for designed proteins, so it's very easy to predict. But for RNA, it's hard. But it's maybe because we haven't trained enough of the network. Okay, so probably we also need to this kind of human intervention for this designed RNAs. But okay, probably the story is too short because we only tested 4 designed RNAs in this experiment. I don't know how would other RNA look like, especially for shorter RNAs that are designed. For these 4, they are too long for AI-based method. I don't know how will the results be for these shorter RNAs. Probably we need more tests to have a pretty clear conclusion. Thank you.
Yeah. And I think all the RNA, the synthetic RNAs, they come from the same lab, probably. Yes.
Yeah,
So there could be some kind of bias or some way how these RNAs are designed, and it's difficult to predict them. There is a new question, so I will start maybe with this one. So for synthetic RNA, so just to close synthetic part, synthetic RNAs, when you mention AI plus motif/fragments based modeling, does that mean focus on making structure predictions for smaller subsequences of RNA? So yeah, the question is if this for the synthetic RNAs would be beneficial to find modules and then
yes,
models. Yeah.
I think that's right. So that's right. For this synthetic RNA, it's very, especially for these big RNAs. So we need to predict some motifs correctly. And also you can see for this synthetic RNA, it's very long, but they have some basic motifs. They have several, probably 7 long alpha helices. So it's very important to predict these motifs correctly first, and then assembly them. I think this is the approach done most of the top human groups in CAS 15 for these synthetic RNAs. But as I said, for shorter RNA, I don't know what the story will be. So we need more tests.
Okay, so now going back to natural RNA. So there is a question from Arni. A question, to both of you. What is the best way to make alignments? Search, infernal, DC align, research, I believe, manual research.
Okay,
What are your thoughts on building alignments?
So, yes, for the MSA, and in fact, I talked about this before. So for this RNA, the MSA is pretty more difficult than protein. And in our test, we find that the Inferno is the most effective one. In fact, to be frank, I didn't test the DC online or other. I test Inferno and also Blast N. In fact, pretty iteratively. Blast N find some homologs. Then after this one, you just try to improve this by Inferno. In fact, there are a few RNA alignment tool published, but the problem is they are very, very slow. They take a very long time to search the big huge database. But unfortunately, after you spend days to complete the search, then you will end up with with alignment that are not necessarily better than the fast search. So in fact, in our server, the trRosettaRNA server, to make it faster, we just use the Inferno search on the RNA central database. So it's very fast, but for most RNA, it can work. So I didn't test the DC align. Sorry, I don't know. I don't know this approach.
Okay, Rachel, do you want to read your question?
Sure. Yeah, I had a question about natural RNAs actually between R1107 and R108. So these are highly sequence similar. They only differ by one, I believe just one mutation. But your initial model, so it was all corrected by the time you used RFAM secondary structure. But the initial models were very distinct. So I was wondering if this was solely just due to spot RNA predicting the secondary structures very differently for some reason, or did your network also treat these kind of single mutants differently?
Okay, Wenkai can answer this question.
We don't treat this syndrome mutation 就是 sorry, we don't treat this mutated sequences. We just think it is two different sequences. Yeah.
So the secondary spot RNA predicted the secondary structures different between the two?
Yeah.
Okay.
We separately predict secondary structure and ran the trRNA course. We don't consider the effect of the mutate.
Cool. Thanks.
Okay, but the prediction by spot RNA it is not accurate for these targets. You can see here, so it's wrong, the prediction. So that makes the 3D structure prediction bad for this target. So by improving this secondary structure, you can see we can get better 3D structure prediction. I think this story is similar to the human group. They are improving secondary structure, then get a better 3D structure. But this information is not available to this AI-based automated prediction.
Okay, so maybe the last question for today for Pranav. Is it possible to artificially generate more synthetic RNA training data from existing synthetic RNA data? So,
yes. So your question for me or for us?
yeah, for both of you, I guess.
Okay. So I think yes, we hope to have some data for these synthetic RNAs. So currently we only have 4 tested in CASP 15. So because for the Rachel death, he has some data on the synthetic RNAs. Especially for this one with smaller size, we wish to have this data to test how the deep learning based method will go. Also, it's also very important to have this synthetic data to train between our network. Yes. So I know the Rachel Desk, so he already has more than 1 million design sequences, but we haven't yet touched that.
Yeah. So I think if you want to learn more, our previous seminar was about the reduce idea, how to generate more data, which is, I think there are lots of synthetic RNAs there. There are natural RNAs. So there's 1 million sequences and a lot of low resolution experimental data. So yeah, probably this is possible, going back to the question. And yeah, if you want to learn more about it, then you should go to our previous seminar. The seminar is on our YouTube channel. This seminar will also be there soon. And I think, yeah, thank you very much for the wonderful discussion and the talk, and see you in 1 week. Thank you again.